Okay, so good morning, everyone, and welcome to another seminar in bioinformatics. As most of you know, this is organized by our research group at Center of Biological Engineering, the Biosystems Group, together with Nebium, the Students Association in Bioinformatics, and also with the collaboration of the directive board of the Master in Bioinformatics. So we have a nice crowd today watching this presentation with both students and researchers from our group, and also some invited guests from Silico Live, which I think will enjoy this presentation. I thank Pablo Carbonell for accepting our invitation to talk today about his work. He has recently moved to the Polytechnic University of Valencia, but he has already a long work in this area of microbial engineering and retrosynthesis, biotechnology in general. And he will talk today about some of the most recent tools in this line of work. So thank you, Pablo, for accepting our invitation. You have the floor.
Thank you, Miguel, for the presentation, and welcome to everybody. I'm happy to be here today to present this talk about designing predictive tools for microbial production automation. As Miguel mentioned, I'm currently located at the University Polytechnica de Valencia, where I moved recently, but most of the work that I'm going to present has been developed in collaboration with centers in France at INRA, and also in Manchester at the Manchester Institute of Biotechnology. Okay, so let's start. So, okay, first I would like to give you just an outline of how it's going to be organized, my talk. So first of all, I will discuss a little bit about synthetic biology. Probably most of you are already familiar with it. But what I'm going to discuss is how it is related to the bioeconomy and the role that is going to play in the new bioeconomy. Then we will see how we are going into that direction through automating the design, build, test, and learn pipeline. Then I will talk about several design tools that we have been building in the last few years. When we talk about design tools, you have to think that it's also, we could also call them bioinformatics tools, no? But you will see that all of them are in the context of biotechnology and metabolic engineering, and how we are developing them into cloud-based approaches. Then I would like to introduce some more like advanced applications using machine learning and dynamic regulation. And I will conclude with several strategies of what is more and more being called the smart biomanufacturing. Okay, so let's start first with synthetic biology. I think most of you have seen this picture on the left, which is just a cartoon, no, that is representing the cell as if it were a computer. No? So, well, obviously, this is probably a metaphor that is going too far away, because what we want to actually is to engineer the cell in a way that we can take advantage of biological processes and probably perform some computation that will not be able to be achieved by using a regular computer. So rather than having here a hard disk and a microprocessor, which probably doesn't make a lot of sense, what we want is to use memory systems that exist in the cell, or biocomputation system that exist in the cell naturally, and maybe has been evolved in nature for some reason, or we could maybe think about them. This is why synthetic biology is defined. This is one definition that is often mentioned. Synthetic biology is the engineering of biology, the synthesis of complex biological-based or inspired systems which display functions that do not exist in nature. So this is the definition that we want to use, but maybe a decade ago, 10 years ago, it was mostly a topic that people were discussing in scientific communities. You could see a lot of journals like Nature Science with covers about synthetic biology, and that was for many years. So there was a lot of excitement in the scientific community, and what we have seen now lately is that actually this excitement is moving more into the industry and economists. For instance, this journal, the Economist, which is a magazine that is read by many people, politicians, economists, not and the wide public. So it's not only for scientists. And they put again synthetic biology as its cover last year, saying that it's going to be the technology that is going to redesign our lives. Okay, so that shows that there is a lot now of excitement about this. And you could see this when you look at the startups ecosystem. Companies that now are being created in small companies usually, but they are getting a lot of funding, especially around the Bay Area in San Francisco. It's like the Silicon Valley, what was a revolution when all of these internet companies appeared, maybe 20 or a little bit more years ago. We have now the same for companies that are based on synthetic biology. So we could ask ourselves, are we the next big thing? And you see every day on TV, people are talking about companies like Moderna. Moderna is a synthetic biology company that works with RNA, or other companies like Samergene, or also it's not only about pharma companies or about chemicals. It's also about, for instance, making food. You see that people now are growing burgers, are growing meat using cell culture. So we can see that there is more and more investment going on into synthetic biology. And I think this is because we are now with a big opportunity that is being created by the bioeconomy, where what you have is that billions of tons of renewable biomass is being produced every day from households and from factories. And all of this biomass, what we are trying to develop are this type of facilities or platforms, which are called biofoundries, where we can transform them using bio-based processes into chemicals with an added value, and in that way to create a circular economy. Okay, so what is usually regarded as waste for us is something that actually has a value, and it can be converted into chemicals that are of high value in the market, usually related to natural products that you can extract from plants, but it's very expensive. And you can make it now by fermenting in bacteria this biomass. However, for many years there was a critical view. People would say that okay, all of this sounds very cool and very exciting, but the reality is that companies are spending a lot of resources in terms of money, in terms of time, human resources, in order to achieve something that actually is competitive, is something that can actually be introduced into the market. And that happened, for instance, with the two main examples that usually are cited about this approach, which is the propanediol developed by DuPont and the artemisinin, which is an anti-malaria drug. In both cases, it took a lot of years in order to arrive, and also many people were involved. For instance, artemisinin, Berkeley, Amiri, Sanofi. but it took a lot of time and resources in order to arrive at a final strain with tighter rates and yields that were industrially ready for the market. So what happens is that now this picture is changing, because there are novel technologies that are appearing and are making easier for a product to be developed in a reasonable amount of time and with a rapid development approach. And this is what we call now the smart biomanufacturing revolution, which is a new way of integrating what is the in silico design, which is more the modeling, the computational part, with the smart biomanufacturing, which is like the smart manufacturing equivalent. The smart manufacturing, let's say, it is a concept coming from the Industry 4.0, from the Internet of Things, that sees a factory, a plant, like a full connected system that can be fully automated, and it is generating real-time data that it can be then processed by automated learning, by machine learning, and then use this information in order to update our in silico design. So you can see here how all of these technologies are converging. For instance, this is a picture that explains what is the current vision. So we have, first of all, these biofoundries where we apply the design, build, test, and learn also can be defined here in order to have a full engineering cycle where everything is underpinned by automation. And we have multiple tasks like experimental design, host selection, host modification, pulse assembly, where all the genes are put together, a combinatorial library is created, and all of these allow us to perform rapid prototyping, which means that in a faster way through several cycles, we are able to develop strains, like it could be a bacteria, it could be a yeast, which are going to produce detectable amounts of our target product, okay? And then this prototype can be transferred into these large fermenters, what we could call the biorefineries, where it's going to be performed the scaling up, okay? Coming first from the lab scale into a pilot plant, then a demonstrator, and finally a production. So these are concepts that in the chemical industry has been used for a long time in the pharma companies, and now we want to have them also for these biomanufacturing systems. The way to achieve this is by automating the full biomanufacturing pipeline. What you can see here is this workflow where all the different tasks that are involved in the design, build, test, and learn stages of the pipeline have been identified, like pathway selection, enzyme selection, DNA parts design, design of experiments, assembly for the design, and then this is we are moved into the build, where we perform the DNA synthesis, the part preparation, assembly, purification, sequence verifications. So what is important here is that first we are able to identify each task, and then to develop standardized protocols, or to develop some automated tool, like it could be a software program that is going to perform the task for us. For many years, all of this was performed in the lab by molecular biologists in a very manual way. So every protocol, okay, there were protocols for each process, but then they have to be performed in a very sequential and time-consuming way. And this is something that we want now to automate using robots, using platforms that allows to perform this type of operations. The same goes for the test, which is more the analytical part, where you are going to look at these cell cultures and see if we can detect the product that want to produce. It could be done by mass spec, it could be done by other techniques. Metabolomics is one of the most common techniques that is used for that purposes. And finally, once we know what is in the cultures, which metabolites and which molecules are present, we can train some machine learning system which is going to inform the design step again. Okay, so we can redefine the design rules that we use in our process. So I would like to say that this concept is very close to the concept of the SCADA system, which means supervisory control and data acquisition, which for many years has been proposed in other domains of the industrial processes. And we would like to have the same approach now in our biomanufacturing environment, so that we can bring all these concepts coming from the industry 4.0, like automation, Internet of Things, big data, cloud computing, and everything can be controlled through this SCADA system that we can access from our computer. This is going now very is becoming quite central to several international initiatives and consortia, like the Global Biofabrics Alliance, which is like an alliance of several biofoundries internationally. You can find them everywhere around the world, and they are trying to develop standards in order to be able to exchange data, to exchange results. And the same goes for other projects like IBISPA, which is funded by the EU and involves lots of companies, or Shiki Factory 100, which is another project funded by the EU with industry and academic partners. And in this project, the University of U.Minho is also involved, as well as recently now the Polytechnic University of Valencia. So as you see, there is a lot of interesting initiatives going on. And now I'm going to explain you in more detail what are these design tools that are so important and how we can develop them in a standard and easy way. So I like sometimes to show you this image here because from my point of view, when you perform the design, or if you were a biodesigner, probably nowadays, if you look into job offers, you won't find a lot of biodesigner jobs, because it's a term that is starting now to become more and more well known. But already you can see small companies in around the Bay Area or around Boston, where the job of a biodesigner is announced rather than a bioinformatician. And what is a biodesigner? You can imagine it's like the work of an architect or a designer, interior designer, for instance, is somebody that has on his or her desk several parts, DNA parts in this case, but wants to combine in a way that creates a big library that allows us to explore all the possibilities of our design space. To that end, the designer can tune the system, like it has different knobs that allows to tune the system, and it has some models that try to explain what's going to happen. And then once we have the design, this design is sent to the cloud, let's say. It's sent to a lab where through automated standardized protocols, the library is going to be built and is going to be analyzed and eventually is going to be scaled up into fermenters. And all of this data that perhaps you are sitting there in Portugal, but all of this experimental part has been performed in China or some other place. And then you will receive the data back, and you can use these big data sets in order to perform the learn part of the cycle, in order to develop new models that will help you to predict in a better way, in a more precise way, the behavior of your system. So that's a little bit the idea. And in order to do that, what we have been developing in the last perhaps 10 years has been a full ecosystem or constellation, if you want, of tools, okay, which are more like apps, like the apps that you have on your mobile phone. Each one performs a very specific task, okay? And what we want is that all of them can connect with each other, can speak to each other through a cloud-based interaction. And in that way, the final user can choose which tools wants to use and which ones wants to actually perhaps use performing a more manual way or using some method that is already present in their labs. Okay, so that's why we are providing a full portfolio of possibilities, like selecting our pathways using this tool Retropath, or selecting the enzyme sequences, the DNA design, the selection of promoters, of the ribosome binding site, the experimental design, or even biosensor design. So that's a little bit the idea. And for instance, I would like to show you one of the initiatives that or projects where I'm also involved, which is the Galaxies in Biocat project. That the main purpose here is to integrate all of these small pieces of software by using Galaxy. Probably you know Galaxy is a well widely used by the bioinformatics community. I think it's a web-based system that allows us to perform a workflow of operations. Okay, usually you use it, for instance, to analyze NGS, next generation sequencing, and so on. But we can use it also in the context of metabolic engineering, and this is what this project is trying to perform. It is now in its beta stage. We have a community of beta testers for the project, and probably soon it's going to be open for the full community. Another example is this animation here, where you see how we use the Retropath tool. The Retropath tool is a tool that we developed in order to search for metabolic pathways that we can introduce into a cell. We have a model of the cell, and then you have a molecule like the one that was drawn, which is a molecule that you want to produce because maybe it's useful as for developing bioplastics or things like that. Then what you have is what we call rules, is a set of reactions that we know that are possible to be catalyzed by enzymes. Enzymes that maybe they are not present in the cell, but we know that there is some enzyme in nature that can be used for this, okay? And then we tell what are the set of precursors that are available in the cell. It could be E. coli, a bacteria, it could be a yeast. And what you get is this huge map here that tells you all the ways of connecting your target molecule to the precursors through the diverse pathways. Okay, you can have sometimes even thousands of different pathways. Each pathway is just a set of enzymes. Okay, and each enzyme what performs is a biochemical transformation, a reaction, which allows to go from your precursors stepwise until you arrive at the target. So, and you can find in the databases the actual amino acid sequence for this enzyme. And then it's a work that is going to be genetic engineering in order to introduce the enzyme. So we want mostly to do this in order to expand the diversity, the chemical diversity that we can obtain through biosynthetic routes. And this is something that you can see in nature. And we want to start from there, but then to diversify more and more the molecules that you can obtain. For instance, here you have a pathway that typically you can find in plants that produces a flavonoid. I put as an example the pinosembrin. And what we want is to transform this pinosembrin into other derivatives, which can be thousands by using glucosylation, methylation, prenylation, hydroxylation. There are some enzymes that are used in order to perform these changes. Okay, so what's going to happen? That we are going to obtain at the end some molecule that maybe has some property that is of interest for us. Okay, and this is something that sometimes can be important, but because maybe you can replace, for instance, a biomaterial by a material that is now produced from fossil resources, like from oil, by a molecule that we have discovered in this large universe of biochemistry. So I'm not going to explain this in detail, but just so that you understand a little bit the way that we perform this type of analysis, which is a bioinformatics or cheminformatics, if you want, analysis, is by taking our molecules and then to see which are the different chemical motifs or fragments that contain this molecule. This is what we perform here for the flavonoid. And as we go deeper and deeper, you see that we arrive at more and more simple descriptions of the molecule. So this strategy can be used, as we do here in Retropath, in order to understand better a reaction and to generalize this reaction as a set of very simple transformations that are focused around the reaction center. So we are just looking at the reaction center, and as you can see here, we can predict many molecules that can be converted by this simple description. When we make it more specific, then we will find less and less pairs of substrates and products that can be converted. And this is the basis of what we perform here in order to predict new pathways. So there is a database that is called RetroRules that contains hundreds of thousands of basic descriptions of the reactions that you can use in order to search for millions and millions of novel compounds that never were found before in nature, but we can predict that they can be possibly or potentially produced by using this enzyme that has this simple reaction rule that we describe here. All of this notation is what is called the SMART notation. That the advantage is that it's very standard, and you can use it with multiple software. So that makes it very convenient, this database, if you want to make predictions. Then we have a software that performs the enumeration. The enumeration is just to look at all the possible pathways that exist in this huge map that you have generated through retrosynthesis. That is this operation that this computation that is done in Retropath. So what you see here on the right are pathways that we found, and there might be hundreds. we have some strategy that is based on elementary flux modes. It's like a way of based on linear algebra of decomposing your big map into a basis that tells you all of these basic routes. For instance, in the Shiki Factory 100 project, this is something that we are using this approach and others because there are several groups, for instance, from Silico Live, from EPFL, that are also applying their strategies in order to enumerate, in order to know all of these hundreds of pathways that can exist. Another tool that we have is in order to annotate these pathways, okay? And annotate, I mean to find sequences that are the actual DNA that you want to express in the bacteria or in your yeast. You have a tool that is called Zellenzyme that is going to predict which are the best sequences that you can engineer in the cell. And here we are just looking at homologies based on the reactions and based on the sequences. So that's one example of producing pinosembrin. We were able to predict 11 pathways that can be used, and they were ranked here using multiple criteria, like which pathway is more toxic or which pathway we are expecting that is going to produce more of the target. And in that way, that was then expressed and experimentally was tested, and we found that actually there was a good correlation between the predictions and the actual levels of production in the lab. So that gave us an idea that this approach could be very useful, because you don't need to try 100 different pathways in the lab, because this is going to take a lot of time, a lot of efforts. Okay, so it's something that you can make it more in a way that allows us to prioritize those pathways that are more promising. So we are applying, as I said at the beginning, these strategies also through machine learning approaches. And this is going to allow us to expand in a more rational way our targets. Just to give you one example. For instance, here, these are monomers that are used in order to make polymers of plastics, basically. You know, this is what you can find in any commercial database. And these points are the predicted property of the polymer. Okay, in this case, is the transition temperature, which is a property that usually is important for polymers. So then, what we did here was to, in blue, to look for all molecules around these monomers that can be produced by retrosynthesis, by introducing biosynthetic pathways. And then using machine learning, we predict the properties of these blue molecules, okay? Well, actually the property of the polymer that is using this basic monomer. And in that way, we can select polymers that now are going to be bio-based produced. So these are like bioplastics, let's say, and they will have enhanced better properties than the ones that are commercially now synthesized. So these are the type of things that we like to do with machine learning. And also, another approach, which is quite nice, is to use machine learning in order to improve our DNA design. So what you see here, this is a library that we want to express on a plasmid in bacteria or in yeast, because it will express a pathway, okay? But this library is quite complex, because you can have several promoters that can be used. You can also change the ribosome binding site. You can change the the actual variant of the gene that you want to use. So the problem is that all of these regulatory parts, like the promoters and the RBS, often it's not easy to predict how they are going to change the properties of your pathway. There are some tools, but often they also depend on the context. So it's not the same in your pathway for producing pinosembrin than in other pathway for producing styrene, for instance. And what we did here was to measure some of these sequences, okay, for the RBS, and then apply deep learning, a machine learning technique like a neural network, in order to predict the properties of the rest of the RBS. And as you can see here, because this is the comparison between the prediction and the actual experimental values, you see that there is a good correlation. Okay, so this system is quite powerful as a predictor. And the same goes for selecting the promoters. We also build a tool that is able to predict the strength of the promoter. So you see that it's just a matter of going into the lab, building a library, sequencing the library, or knowing which DNA you have there, and then applying some techniques like a deep learning in order to make a predictive tool. We did the same for the designing biosensors, which actually, in our case, well, I'm not going to explain in detail, but basically what we look at is for molecules that can be detected. For instance, quercetin is a molecule that there are transcription factors, like small proteins that can bind to quercetin, and in some way they interact, and this can be used in order to regulate the transcription of a gene. So you can use it as a reporter, so that it tells you the presence of or not of quercetin. But through several steps, we can convert other molecules like pinosembrin into quercetin. And this is what this tool tries to do, to find ways of having a biosensor for any given target. And the tool is called Sensipath, and it's available as an online tool. And recently we use it in order to build a full dynamic regulation system for a molecule, like in this case is the naringenin, which is a molecule, another flavonoid. The naringenin can be converted into some other molecule, which is the camphorol. And camphorol is a molecule that can bind a transcription factor. And in that way, we can regulate the levels of expression of a gene, which is the CHS, that is the chalcone synthase. That is one step in the flavonoid production. So by detecting the level of this molecule, we can regulate, therefore, the actual production that we are going to have. So it's like a feedback loop, like you can see this in this schema. In order to make these feedback loops, it's important to have biosensors, which can be based on transcription factors, or they can be based on RNA through riboswitches. Okay. Well, you have seen many pictures of these combinatorial libraries. In order to make them the designs as optimal as possible, we need to apply some principles from statistics that are what are called statistical design of experiments, so that they are not just created randomly, but they are created in a way that allow us to learn as much as possible. For instance, in this paper, what we did was to create this initial library, which is optimized. Okay, the combinations are not random, but they are optimized. And then we measure the production of the molecule. And once you selected the actual molecule, once you select the one that is producing the highest amount of the actual molecule, you can use this in order to learn some rules. That's what you see here at the bottom. And then build another library that is more efficient and brings you to higher amounts of the product. And we can do this in a very nice way. I mean, if you see here, this is the comparison between what is predicted by this model and the levels that we were obtaining in the actual library. So you can see that there is a good correlation for most of the cases. Obviously, some cases might happen that maybe there were some point mutations or things like this. So not always is perfect, but we have a nice correlation, and that shows us a proof of concept that is possible to perform this type of approaches. Okay, you have seen a lot of tools, and now just to conclude my talk, I want to show you one example where we applied the full pipeline with all of the tools in order to accelerate the production of new, of novel biosynthetic routes and a set of target chemicals. So the idea that was performed last year in Manchester was to apply a stress test to the SynBiochem Center, which is the biofoundry there in Manchester, in order to see how many monomers, which are molecules that are used for materials, for building materials, how many of these monomers were possible to produce in a short amount of time, just in a time frame, or within a time frame of 3 months, how many we can produce. Well, here you have the full list, and we made a very manufacturing approach in order to implement this project. So, for instance, we have these types of time schedules where you can see for each project when how many days was at the design stage, at the build stage, at the test. So here, well, you can see all of these steps are the one that at the beginning of my talk I showed you on this large workflow. Okay, so you can see in parallel how for each project is working. The ones that are in red are the ones that we were not able to produce, because obviously you have a limited time frame. Not all the projects are going to be successful, but all the ones in red where we were able to produce them. So we built like a large map of new molecules, all the ones in green that expanded the portfolio of molecules that can be produced in the lab. And in some cases, like the mandelic acid, it involved also other things like, for instance, directed evolution, fermentation. At the end, we were able to obtain productions above 1 gram per liter, and therefore already close to industrial levels. So I like to put here like a summary numbers of what happened during this almost 90 days. So where it was possible to build 160 genetic parts, 187 plasmid pathways, 142 enzyme assays, also 9 mutant chassis, 8,000 samples were analyzed by mass spec, and 22 new methods were developed for mass spec. And in total, 12 of 27 targets were produced in vivo, and they cover 5 of the 7 classes of material monomers that we initially were targeting. So I think these numbers are quite impressive, and this is something that, as I said at the beginning, 10 years ago or 5 years ago, it was not possible. People were spending 10 years and a lot of money in order to, and resources in order to be able to just to produce one molecule. Now, in 3 months, we are able to produce 12 molecules, and the resources that are required are less, and we can be very quick in our development. So that was possible through several scale-up strategies, which I already mentioned, like directed evolution, optimization, fermenter optimization, strain optimization, media optimization. So and the titers were up above 1 gram per liter. Okay, so that's what I wanted to show you today. And just as a take-home message, as you have seen, biomanufacturing is ready for the demands of the future bioeconomy. And biomanufacturing is engineering, but it's also bio. That is, requirements can change during the course of the project. So we need to be very smart. That's why I like to say smart biomanufacturing in the way that we design these processes, because there's a lot of challenges. And that's why people generally say that we need to have an agile approach so that we can readapt as we find new challenges, and also cloud biomanufacturing. So at the end, what is enabling all of these tools is automation. So automated design is the one that allows correcting issues at the earliest point in the process. So that right from the beginning, we're already thinking on this large scale process that at the end we want to implement. Okay, so that's what I think the main conclusions. Just mention that the acknowledgements of the University of Valencia, of Manchester, and the project Chiqui Factory, and also the SynBio Control. So thank you for your attention. And also, if you are interested maybe on some master internship or PhD in the future, please contact me, and we can think of possibilities of doing this at the University Polytechnic Valencia in my lab, or maybe in collaboration with your institution. Thank you very much for your attention.
Okay, so thank you, Pablo, for this very nice presentation.